Why not to learn something, hopefully something new today. So this presentation is going to be focused on the research that we currently have here at OSU for my PhD program. And because of the short time that I have, it's going to be just a brief, short presentation about everything that we are doing here. And also because it's kind of late, it's going to be short and sweet, and hopefully everyone will enjoy. So first, to start with a quick overview or a quick information, it is widely recognized that mycorrhizal fungi, or AMF, is an important component of ecosystem dynamics, as they are viewed as the absorptive root surface outside of the nutrient depletion zone of the plants. So even though that there is a lot of research with mycorrhizal fungi, there is still a lack of understanding on how such effects occur in rangeland plants and how we can use these for other purposes, such as ecological restoration. A lot of this happens mostly because the majority of the research that we have is currently focused on crop plants. And croplands, in many cases, they differ in life cycles and life strategies. There is very little work on rangeland plants. And in many cases, this has resulted in contrasting results and makes a clear signal that we really need more research on AMF, and with a special focus on rangeland plants that eventually could be used for ecological restoration of degraded ecosystems. So this was just a very quick overview of why we are working with this. And the general objective of this study, or the work that we are conducting here, is that we want to understand the rangeland plant benefits to AMF that eventually could be used for restoration programs. And for this, we have developed 3 consecutive experiments. First, we wanted to understand how Artemisia species respond to commercial inoculum and AMF present in an early sterile soil. And after that, we wanted to see if Artemisia had hidden benefits to AMF to make this plant more competitive against invasive species. And finally, we wanted to understand how different sources of inoculum could produce different responses associated to different plant species with different plant sterile status, comparing, for example, early sterile plants with late sterile plants. But working with AMF is not something very simple. I have seen that a lot of work is done without properly understanding many of the variables associated with fungi. So first, we did some preliminary work, first to try to learn more about mycorrhizal fungi. Because we were using a commercial inoculum and we wanted to see if the commercial inoculum could produce an effect on the plants. First, we needed to see if the inoculum was actually viable or not. So before conducting any work, we test the commercial inoculum viability. And since the best way to understand AMF is by plant colonization, we did this by growing crimson clover with AMF inoculum for 6 weeks. Plant species or clovers, because clovers grow relatively fast and they are unknown mycorrhizal plants. So clovers were grown individually on containers with either autoclaved soil or autoclaved soil with the inoculum, just to see if the inoculum was able to colonize the plant species. So it was just a preliminary work. And also, we also studied, because all the work that we did was with seeds, we also tested the viability of all the seeds from all the species using tetrasolium in the seed lab from Oregon State University. As it's important to know first if the seed source is able to produce healthy plants that eventually could grow into what we call a normal or a healthy plant. So after knowing that the inoculum was working and the seeds also were alive, we started with our first experiment in which we grew low sagebrush, Wyoming big sagebrush, and black sagebrush together also with medusa head on autoclaved soil plus inoculum, and early cereal soil, and an early cereal soil plus inoculum. And with this, we were measuring the ability of the plants to be colonized by the inoculum or the fungi present in the soil. And also we want to see the differences in terms of plant biomass of all of these species with these soil treatments. So the plants were grown for 15 weeks in greenhouse conditions. And after that, we analyzed the roots using the clear and stain method with trypan blue. And eventually those roots were evaluated with the line grid interception method to count for colonization on the roots. And just as general results for this first work or experiment, we found that, of course, the inoculum colonized the clovers, but failed to colonize our target species. So all the colonization of these plants was restricted to plants grown in the early cereal soil. So the inoculum, for some reason, was working, but it did not colonize our plants. And the main response that we observed was a growth depression at the end of the growing period. So the plants that were colonized for some reason were smaller, which was the opposite of what we were expecting, because we expected bigger plants in this study. However, the same happened with the invasive species. So in this other picture, we can see that medusa head grew better in the autoclaved soils, which indicates that a biological component present in the soil was reducing the growth of this and all the other plants. However, because we failed to incorporate a microbial wash to reintroduce the soil microbes back into the soil, we have some limitations of this study because we also believe that this response can be two main things. One, that we have pathogens on the soil, or that the plant was actually paying more carbon to the fungi before obtaining a benefit. So we were unable to see that separation just because of that. That was something that we learned because of this. And in this other graph, you probably can see with a different idea the general response of these source species to the treatments after 15 weeks. So here with big sagebrush, we can see that the two soil treatments that were autoclaved soil or the autoclaved soil plus inoculum produce plants with more biomass compared with the two treatments that have the early cereal soil or the early cereal soil plus inoculum. But because we know that the inoculum did not work with any of these plants, the difference was something that was present in the early cereal soil. The same response was really clear on medusa head. In black sagebrush, the response was really, the plant was really not responsive, but we found a slight increase in biomass in the soil with added inoculum. And in low sagebrush was something similar, but was the opposite. So the plants did not respond very well to the treatments. So we actually believe that this particular inoculum also had another component rather than just the mycorrhizal. So that was the general results of this first study. For the next study of the competitive ability of the plants, this study is still in progress. We wanted to see if the mycorrhizal fungi could enhance the competitive ability in terms of biomass production and nutrient acquisition when exposed to competitions such as drought and competition from invasive species. For this study, we decided Wyoming big sagebrush, black sagebrush, and low sagebrush. The treatments that we have for this study were medusa head as competition with or without, drought as another stressor. We incorporated here again the early cereal soil as a source of inoculum, the commercial AMF inoculum. We just selected a different one for this study as we did not have good results in the first study. And also we used the same inoculum sources, but they were autoclaved just to incorporate a microbial wash in order to separate the effects of the mycorrhizal from the microbial community in this particular study, as we learned something different in part of the limitations from the first study. So the plants growing in competition were grown for 6 months to better see those responses in the first growing season of the plants. Currently, those plants are being collected right now. So the growth already finished, but I am currently collecting the plants. And for this, I still also have some preliminary results. So this is the graph of the growth of big sagebrush over time with the different treatments. So these first treatments are the plants grown without any type of competition and without drought stress. So the plants were growing in, let's say, good conditions. The plants growing with drought without competition are smaller, but all the plants with competition are really small. So those plants are really struggling to produce biomass. And because this has been seen in other works, we believe, or at least we expect, that those plants may have some benefits that are not in terms of growth, but rather in terms of nutrient acquisition, which is one of the main benefits that are expected from plants growing with mycorrhizal fungi. So currently, I am still collecting the data for these experiments. After we obtain all the data and we conduct the biomass analysis, the colonization measurements, and the nutrient analysis, we are going to be able to better understand if those plants were more or less competitive when they were colonized or uncolonized by the mycorrhizal fungi. But still, well, that will be very important to understand because as we can see, Methuselah was able to grow a lot of biomass. Everything that we can see in this first picture is the root biomass of all the Methuselah growing together. As you can see here, the poor big sagebrush was really struggling to obtain light, moisture, and nutrients as Methuselah was really dominating in this particular experiment. So after conducting all of these analyses, hopefully we can have more useful information in how mycorrhizal fungi can benefit this plant that eventually could be used for restoration programs. And the final study that we're still conducting in our greenhouse, we're trying to see if an early cereal soil will benefit more early cereal plants and a late cereal soil will benefit more a late cereal plant. Also, we already work with biochar and mycorrhizal fungi, so we are expecting that if we incorporate biochar to this study, we may have an increase in colonization responses to these plants. So this study is currently in progress in the greenhouse. For instance, we are evaluating blue bunch wheatgrass, ventenata, and Methuselah. Those plants are currently being grown from the inoculum, early cereal, late cereal, and the commercial inoculum from different sources to try to better understand those responses and trying to see which one will produce best results for each plant species. And of course, for the perennial plants such as blue bunch wheatgrass, we are expecting to find best results in the plants grown with the late cereal inoculum source compared with ventenata and with Methuselah that we expect to find the best results in terms of less biomass when grown with these treatments of late cereal soil as an inoculum. And those plants are still being grown in the greenhouse. So we are going to be keeping those until the end of this fall. And for this experiment, we selected grasses because those species grow relatively fast and we can see the plant responses on the first grow stages easily and trying to understand the responses of a wider variety of plant species. So because I am short of time, so some general conclusions is that we really have observed the importance of inoculum viability testing before conducting any study, because working with mycorrhizal fungi is it's complicated. So we're required to start from early, step by step, to try to reduce the risk of a failure in our experiments. Also, because with this type of work, takes some time to really see the responses. 6 weeks can give you some results, but the more time, the best for some plant species. And also it's important to understand that in some cases, the benefits to the plants may not be really seen as an increase in growth. Sometimes those benefits may be hidden or not easy to see, such as an increase in nutrient acquisition or competitive ability. But to properly understand this analysis, we will need to have a specific study design is to target that. But in order to target for those benefits, we first need to understand if those plants are responding to the inoculum and how, in terms at least of biomass production. Then we can jump on the next stages of understanding what's happening with these plant species in a greenhouse and further in the field. And especially because restoration programs, we know that they are tricky, they take time, they can be expensive. But before conducting works with these plants in the field, it's important to first understand the responses associated to the fungi. Because in many cases, we apply mycorrhizal fungi without properly understanding those effects. And in some cases, we don't really see, how can I say this? We don't really see, or we don't really know what's happening there. It's recommended in many cases, yes, apply fungi to these plants to increase their growth, because we know that fungi is good. We really don't know what's happening there. We don't know if the inoculum is actually colonizing or if there is something else in the soil doing this. So there is still a lot of work that can potentially with this field of study. So hopefully after finishing these other experiments, I can have a better understanding of what's happening here that eventually I can use for studying more and restoring some, to be used for using this in some ecosystems. So I feel that I still short of time, so I think I am over or finished here. So I hope that was clear. Thank you very much, and I am open for questions. Thank you.
Thanks, David. That was great.
Thank you.
So I'm going to start out by reading through the chat some questions for you that a couple of audience members asked. And then once I get through those, then floor is open for people for talking and asking questions there. So Alex Yee observes, perhaps a future experiment could use multiple AMF species. Maybe only using one or a few species doesn't confer all the effects seen in nature.
Yeah, that's something that I also would like to see in the future, because I also believe that probably one species can produce some benefits more than others, or probably the combination of some species can be beneficial for some other target plants. But right now, I still feel that this is complicated to properly understand, because obtaining one single species of AMF is not easy. Sometimes we find commercial inoculums as a quick source of inoculum, and they may say that we have only one species, is this one. But really, we really don't know, because we don't see the spores. It's different than working with plants. With plants, we can see the seeds. We know that how the seeds are, but with spores, requires a lot of training to see them and try to understand, or try to see by morphology the species or by a DNA study. But this also can be very important or very useful in the future, rather than using everything together as a single biological community. But that's a really good form of work that can be done.
That's really interesting. Yeah, it's just, I start thinking about all the, like when you're talking about inoculum for certain species and things like that. And it gets very complicated, doesn't it? And so we have another question from Haley Mankey. Says, great presentation, very interesting. I'm wondering why you picked the species of rangeland plants that you did, specifically Medusa head.
Oh, that's also something that was kind of personal, and also because we had some work with this. So Medusa head, I choose this plant because when I first learned about this plant, I was learning the plans for the plant ID contest of the SRM. And I was wondering why they had chosen that plant that for me was not a big deal. And then I came to Oregon and I understood what was the issue with Medusa head and that there was a lot of work with Medusa head. So I became really interested in what was happening with that plant, especially because I learned that in the native soil of this plant, the plant was not a very big deal. And with sagebrush, I also started to really like sagebrush when I was learning the plants. And then I saw that for black and low sagebrush, we had a very few research. So I started to become more interested about what was happening with other plants. Because for big sagebrush, there is a lot of work on many things, but it seems that we focus on the most important plants and then we start to forget about other important plants in these systems that can also be very important. Now, probably we can find lots of important uses. So that's why I choose those plant species.
So Alex, you actually had a comment to your first question. I dimly recall that only having one or a few species creates a kind of monopoly situation, triggering a more pathogenic pattern of activity than when competition is present, or I could be completely misremembering. Alex, if you have a mic, you're totally welcome to use it, by the way. But I'll read out your comments all day if you want.
Okay, thank you. We have more questions or comments. That's a very interesting comment, trying to see the pathogenic effect of happening in the soil. Well, originally, we were not considering the possibility of having pathogens in the soil, but after understanding that we were using an early sterile soil that is present in a disturbed ecosystem, we may have pathogens there. So there is that makes opens more possibilities for research because pathogens are also present in the soil and they also could be influencing the response of plant species in the field. And one of the reasons for using an early sterile soil was that we did not want them to make more disturbance when using soil. And also, another idea was that when we are restoring an ecosystem, we are not going to be restoring a climax ecosystem because that one will not require to restore. An early sterile ecosystem or a disturbed ecosystem is the one that will require to be restored. So understanding more the responses in this type of soil, we believe that is very important, at least for this type of studies.
And so maybe can you describe what an early cereal soil is versus a late cereal soil? I know that certain plants will grow, but I mean, there's probably different layers in the soil at different depths, and I guess the richness would also differ.
So for this early cereal soil was obtained in a place that was lacking a lot of the native plant community. And we also believe that ecosystems that don't have the native plant community or the perennial plant community will have a different mycorrhizal ecosystem. So this soil was from the Crooked River, the national forest. We had a national grasslands. We had a permit to collect soil from there, but the idea was to have the less amount of disturbance when obtaining this. So the soil from this ecosystem, imagine an ecosystem that the native plant community is mostly crested wheatgrass, sandbird bluegrass, and also Medusa head. So we don't have a lot of perennial plants in this area. The late sterile soil, we took a small sample from an climax community close to Bend in which we had big sagebrush, we have spinning hop sage, and other plant species in which was a clearly undisturbed ecosystem, in which we were expecting that the fungal community here will be more adapted to plants ecosystem. So for the last study, that's what we tried to see, if the fungi from a climax community could benefit a climax plant species. And we could have used many other plant species for studying this. But also I like to work with many plants. It's like, we like to say in our lab, like diversity is the spice of life. So we like to use plants here to understand what's happening in the community, because at the end, a rangeland is a really dynamic ecosystem with many plant species, not just one. So by learning the more plant species that we can, it's going to be beneficial for a lot of work, for a lot of opening many opportunities for the future.
Thanks, David. So I'll give a last call for anybody. It's ending off really perfect. We're right on time. And so if anybody else has any questions, we have time for one or two more, but if there aren't, then we'll close out.
I'd actually like to contribute a little bit more.
Sounds good.
All right, so I was actually thinking more in the line of the arbuscular mycorrhizal fungi species in the absence of competition from other AMF. I was thinking more along the lines of them being flexible and finding a pathogenic lifestyle more beneficial than a mutualistic one in the absence of competition. I don't exactly remember the original source I found this from, and might be wrong. It's like with carnivorous plants. Some carnivorous plants only show carnivorous activity in low nutrient environments, but when taken out of low nutrient environments, say you plant a Venus flytrap in a garden soil pot, that they stop being carnivorous and act more like regular plants. So I was thinking maybe these fungi perhaps don't act as mutualistic as we observe in nature due to lack of competition.
That probably can be, that's interesting. I will need to read more about that, but I also know that, or is expected that when we have, for example, the mycorrhizal fungi colonizing the plant, and if we have, for example, like you mentioned, that an ecosystem with a lot of nutrients, the plant will not have, the fungi will behave or respond as pathogenic, because the plant in this case don't need fungi because there is plenty of nutrients. So the plant, if it's colonized, it will be paying carbon to the fungi to keep the symbiosis, but it's not really getting a benefit. So it's just paying out of nothing. So I don't know if that's probably the same idea, but it's also worth looking more, try to understand more what's happening in these scenarios. So thank you for that comment. I will keep that in mind to learn more.
All right, so we are right at 7:30 here. Well, 7:31, but so I'm going to call it a night for everybody today. Thank you so much, David, for taking the time to tell us about your project. And, I'm really interested to see how this is going to go moving forward, because you're talking about how you're going to do nutrient analysis and actually measuring biomass. So yeah, if you want to come back and tell us about your results, and yes.
Thank you.
Most welcome.
I actually plan to talk more in the Rangeland, in the SRM meeting that we have in February. I will be focusing more on the first study, because for that first study that I described, we already are working on a paper for publication. So I want to focus more on that one, as I feel that as the SRM time comes in, I still will be analyzing all of the plants that I have in the greenhouse. So because I already have else about the first study, that's the one that I want to focus to talk more in the SRM in the future.
Cool. Well, hopefully we'll hear you at this meeting coming up, or this tour, AGM.
Oh, yeah. Sorry, what?
Sorry, I probably cut out. This AGM that's upcoming, you'll be in this one. You'll be presenting.
Which one?
Oh, Society for Range Management. You were talking about. You were going to present at the AGM.
No, it's for the Society of Range Management, the annual meeting in February.
Yeah, you're going to present this year. Well,
Yes,
next year technically.
Next year in February, that's the plan. Yes.
Yeah. Okay, cool. Well, hopefully we'll see you there. And so I guess that concludes our webinar for this evening. Thank you everybody for attending. And I just want to set a reminder for everybody that next Tuesday we have another webinar at 12 at lunchtime. And that's going to be Dr. Tim Paulson. He's going to be talking about the history of range management and looking at history and moving forward with our current range ecosystems that exist. And so with that, everyone have a good evening, and we'll see you next Tuesday.
Thank you.